Welcome to your Anatomy and Physiology, Unit 3 exam review. And like always, your exam review can be found in the Dr. Higgs tips section. And when you click on it, it's a little Word document. Does the exact same thing like the past exam reviews, go chapter by chapter, bullet point by bullet point, letting you know what to focus on for the exam. And for Unit Exam 3, luckily it's only 2 chapters. But because it's 2 chapters, you got to make sure you know all these bullet points in detail. So take your time when studying. But again, some of this we know from lecture because it's just muscular tissue or muscle tissue and nervous tissue and nervous system. We've seen these tissues already, way back from when we did tissues. Remember, this class repeats itself a lot. And when it comes to tissues, tissues tend to keep coming back as we jump from organ system to organ system. So when we jump to muscular system, yeah, your 3 types of muscle tissue come back, and that's a bullet point in your exam review. And when we get to nervous system, yes, nervous tissue comes back with the neuron and its neuroglial cell. Remember, those were the 2 cells. So tissues come back, and we'll kind of revisit it a little briefly because we've seen it a couple times now. You've already seen these tissues when we did tissues in lecture and when we did tissues in lab. Now you're seeing them for a third time when we see them here. But again, just going to go through this, kind of focusing on the topics that tend to kind of confuse students and trip them up on exams. So let's go through this. Some of this, again, you already know. For example, when we get into chapter 10, I mentioned tissues come back. It's the same tissues. Okay? So again, I'm using lecture PowerPoints. They're still good. We're going into chapter 10. Remember, we have 3 different types of muscle tissue. Remember, there's skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. This one slide kind of summarizes everything we know. Remember, both skeletal and cardiac muscle tissue are striated. You're going to see those alternating light and dark bands. Remember, we call those striations. And in this chapter, you find out what makes up those striations. What else? Remember, your skeletal muscle tissue had multiple peripheral nuclei, meaning each cell has multiple nuclei that it's pushed to the edges of the cell, to the periphery of the cell. Remember, in cardiac muscle tissue, cells typically have 1 nucleus, sometimes 2, but usually just 1. And your smooth muscle cells only have 1 nucleus per cell. You go, and there's other differences. Remember, your cardiac muscle tissue, when you look at it, every once in a while, it'll look like there's a really dark striation. Remember, that's not a striation. Remember, those are intercalated discs. Remember, they are part gap junction that's allowing cells to send that electricity and that information from one cell to another practically instantaneously so that you could get the heart to beat as one. Because you remember, you find cardiac muscle tissue in the heart. And you can also see another difference in this picture. Looks like these cells are splitting. Remember, we call that branching. You see branching cells in cardiac muscle tissue. And remember, smooth muscle tissue is called smooth because it's missing the striations. There are no striations in smooth muscle tissue. It's non-striated. And then where do you find the other 2 tissues? Remember, you find skeletal muscle tissue and skeletal muscles, the organ, which helps you to move. And remember, you find smooth muscle tissue in places like hollow organs. Your intestines is an example of a hollow organ, and it's also helping things to move. And remember, there was one last important characteristic of all these muscle cells. Remember, you don't control all of them. Remember, you only control skeletal muscle tissue. Remember, when you control the tissue, we say it's voluntary. But you don't control cardiac or smooth muscle tissue. They're both involuntary. So that was just a quick summary on your muscle tissues. Here we go. And then we gotta eventually get ourselves up to a muscle. We gotta build a muscle. And to build a muscle, you gotta get a whole bunch of muscle cells. And you got to hold those muscle cells together with connective tissue. You got to know what I call your connective tissue coverings and what do they cover. There's something called perimysium, endomysium, and epimysium. These are all connective tissue coverings. It's just a matter of what are they covering. So first up there is a connective tissue covering around a single individual cell. Here's how you spell it. It's called endomysium. This is just the connective tissue surrounding an individual muscle cell. This is not the same thing as the plasma membrane. This is more outside, more superficial to the plasma membrane. So around one cell is endomysium. If I want to build a muscle, I got to get multiple muscle cells together and hold them together. You hold multiple muscle cells together with perimysium. A collection of muscle cells is what we call a fascicle. Oh, that sounds familiar. Sounds a lot like when we were making a nerve, and you have to hold it together. Remember, around an individual neuron's axon is endoneurium. Around a collection of axons, a fascicle, which is really just a collection of cells, is perineurium. And around the entire nerve is epineurium. This is following the same pattern, but just for muscle and muscle cells. Around an individual cell is endomysium. Around a collection of muscle cells is perimysium. And around the entire muscle is epimysium. Keep it that simple. And then we look inside of a muscle cell. Remember, this class, things could have multiple names. When it comes to a muscle cell, there are synonyms. You could say muscle cell, or you could say muscle fiber, or you could say myocyte. They are all synonyms for a muscle cell. You could say muscle cell, muscle fiber, or myocyte. They are all synonyms. Okay, so your textbook, you see it uses all of them. It'll say a muscle cell at sometimes it'll say a myocyte sometimes it will say a muscle fiber. They are all synonyms for a muscle cell. And we got to go and break down the anatomy of a muscle cell. But again, you know some things already. It is a cell. And you know way back from chapter 3, basic parts to a cell. Remember, all cells have a plasma membrane holding everything in. Same thing for a muscle cell. It has a plasma membrane. And that plasma membrane holds everything inside. Remember, everything inside of a cell is called the cytoplasm. Well, muscle cell has everything inside. It has a cytoplasm. And it has specialized little machines carrying out specialized functions. It has organelles. It's just that you're going to notice, like usual in anatomy, names change. And we give fancy names to certain parts of a muscle cell. For example, I mentioned it has a plasma membrane. It has that outer boundary. We call it the sarcolemma. And it's holding everything inside. It has cytoplasm. That's everything inside. We just call it sarcoplasm. It has fancy organelles like an endoplasmic reticulum, but we just happen to call it the sarcoplasmic reticulum. If you're noticing the pattern, they all begin with the prefix sarco. That relates to muscle. All right. So if you ever see a prefix sarco, you automatically know you're dealing with something in or having to deal with a muscle or a muscle cell. But then we got to focus on the organelles and structures unique to a muscle cell. Organelles we don't find in other cells, like this here. It's called a myofibril. A myofibril makes up a lot of the muscle cell, makes up almost 80% of what's inside. A myofibril is what we call a contractile organelle. They're these little rod-like structures you see inside of the muscle cell. Here's a muscle cell. They kind of peeled back the sarcolemma, aka the plasma membrane, so you can see stuff inside. And you can see it's a lot of these long rod-like structures. Those are the myofibrils. They are what we call contractile organelles because it's the organelle where you're going to see the contraction. To contract means to shorten. So the way you move really is because your muscles are pulling on your bones. And the way your muscles pull on your bones is your muscles are shortening. They get shorter. And when they get shorter, they have to pull on the bones that's attached to them. And they get shorter really because of the cells. It's the cells that contract. They contract really because of the myofibrils. It's the myofibril that will contract. It's the myofibril that will do the shortening. Okay. So when you go to the gym and you lift up some weights, you lift up a dumbbell and you do a bicep curl, you're pulling on your bone to do the bicep curl by contracting your muscle cells, mainly by contracting your myofibrils inside the cells. Okay, so we call it a contractile organelle. And it's going to be filled with contractile proteins. The reason why people eat meat for protein is because meat is muscle, and muscle is made up of a lot of proteins. So when we look at a myofibril, we're also going to have to look at the proteins inside. We'll do that in a second. But again, let's mention the rest of these organelles. I mentioned it has an endoplasmic reticulum, but we call it the sarcoplasmic reticulum. And it's not broken up into a smooth. So it's not making proteins. It's not detoxifying the cell. It's not making lipids or fats. It has its own function. I want you to know the function of the sarcoplasmic reticulum is simply to store and release calcium. You'll see as we go through this and we talk about a muscle contraction, calcium plays a huge role in skeletal muscle contraction. And so you need calcium around a lot, and you keep it stored in the sarcoplasmic reticulum, and when you need it to move, it will release it. Keep it this simple. Sarcoplasmic reticulum stores and releases calcium. And one last thing. Sometimes the plasma membrane, aka the sarcolemma, dives deep into the cell, with a deep invagination or deep inward extension, meaning it dives in. Kind of think if you have an any belly button, if your belly button is called an any, that's kind of like an inward extension. It's diving into your body. Well, the cell has inward extensions of the sarcolemma, of the plasma membrane. We call them T2s. T is short for transverse. We call them transverse tubules. They'll play a role in contractions we'll see later. All right. So these are some unique organelles. One last thing, one more. Your sarcoplasmic reticulums are all these blue things you see in this picture. And your sarcoplasmic reticulum is a large network, but it does have ends. We name the ends of a sarcoplasmic reticulum. It's called a terminal cistern. It's the end, so it's the terminal. Okay. This is just the end of a sarcoplasmic reticulum. It turns out a T-tubule loves to wedge itself in between neighboring terminal cisterns, in between the ends of neighboring sarcoplasmic reticulums. And we give this whole structure a name. Okay. This structure of T-tubule sandwiched between two terminal cisterns. It's called a triad. Triad for three because it has three parts. It's kind of like an Oreo cookie where the cream in the middle is the T-tubule and the two cookie wafers are the terminal cisterns on either end. We call that a triad. Again, it's going to play a role in a muscle contraction. We'll see it later. Right now, just letting you know these things exist. But then I mentioned we're going to have to go inside a myofibril and look at all the proteins inside. It is a lot of proteins. And you know by now in anatomy, when there's a lot of something, what are we going to do? We're going to classify them. When we look at all the proteins, we could classify them one of two ways. You could classify them functionally based off what they're doing, or you could classify them structurally based off how they look. When you look at the functional classes, there's three of them. They're contractile proteins, regulatory proteins, and structural proteins. Like always in this class, names are here to help. So their names kind of give you a hint as to what these proteins are going to be doing. For instance, contractile proteins are going to be able to do the contraction. They're going to generate the muscle tension. They're the ones responsible for doing the shortening. Then there's regulatory proteins. They regulate when the contraction can occur. They dictate, meaning they regulate. They control when you could contract. And this is very important for skeletal muscle tissue because remember, you control it. So you don't want skeletal muscle tissue contracting without your permission. Otherwise, you'll be walking and moving around without any control. So to partially help you control when you do your movement, you have regulatory proteins. And then there are some proteins that provide stability, structural integrity. They provide structural support, so we call them structural proteins. Okay. So let's go through. What are these proteins and what do they look like? First up are the contractile proteins. What proteins are responsible for getting you to contract? It's two of them. It's called myosin and actin. Myosin and actin are contractile proteins, functional. Then there's the regulatory proteins. That's troponin and tropomyosin. They regulate when myosin and actin could come together to do the contraction. So troponin and tropomyosin are the regulatory proteins. And then you have structural proteins providing support, like titin. That's a structural protein. It stabilizes all the other proteins by providing structural support, basically as acting as something for the other proteins to lean on. So those are the functional classes. But I mentioned you could also classify them structurally. Turns out the contractile protein myosin is really thick looking. When you look at it, it looks thick. This big red globular structure, this rod-like structure with all these little globular heads, this is myosin. While actin, troponin, and tropomyosin are really tiny. They're very thin. Like in this bottom picture, all the little purple beads are actin, the little blue line is tropomyosin, and the little yellow thing is troponin. They're all very thin and small. So this affects what we call them. Because myosin is so big and thick, we call it a thick filament structurally. The actin, troponin, and tropomyosin are all very small and thin. We call them thin filaments structurally. And titin actually has a little bit of elasticity. So it can resist stretching, and if it happens to possibly stretch, it could possibly come back to its normal size. I remember that was elasticity. So we say this is an elastic filament structurally. So those are the different proteins inside. These are different proteins inside of your myofibril, inside of your muscle cell. And they're all located in a specific area within the myofibril. It's called a sarcomere. If you were to look at a myofibril, it's a long rod-like structure, and it's made up of these repeating units. We call them sarcomeres. So this little box is kind of outlining an area of a sarcomere. And the sarcomere is called the contractile unit of a muscle cell because it's really where you're doing the contraction. So we say, yes, the muscle contracts, but now you know it's really the muscle cells contracting. And we say it's the muscle cell, but you know now it's really the myofibrils contracting. And we say it's the myofibrils, but now you know it's really the sarcomere with all those contractile proteins in it that's doing the contracting. So because of that, you got to know what your sarcomere looks like. Your sarcomere has proteins in it. Here we go back to the picture. Let's go back to a picture of a sarcomere. Here's a picture of a sarcomere. Okay, tells you in the picture. And you see the protein. You see the thick myosin filament here in the middle, and you see the thin purplish-looking actin, troponin, tropomyosin area. This is your sarcomere, and this helps explain something you see under the microscope. To keep it simple, imagine this is just skeletal muscle. We're looking at an example of skeletal muscle. And remember, skeletal muscle has alternating light and dark bands. It has striations. When you look at striations, you're really seeing where you have a lot of thick and thin filaments. And we name these areas in a sarcomere. For example, there's an area where you only have your thin filaments with no overlap of the thick. That's what we call the I band. But then there is the area where you do have the thick filament, and yes, there is some overlap with thin, but this area stretching from the tip of the thick filament at one end to the tip of it at the other end is called the A band. So when you look at skeletal muscle tissue under the microscope, you're really looking at alternating A and I bands. Those are the striations. Keep going. Let's break down some of these proteins. Oh, there's this zigzag looking structure here. That's a protein. We call it the Z-disc. Turns out that's the borders to a sarcomere. So we say a sarcomere stretches from Z-disc to Z-disc. So this little area in between is one sarcomere. And if you notice, this Z-disc happens to cut the I band in half. So when we look at a sarcomere, how much of an I band do you have? How do we say it? Do we say you have two halves of an I band, half on either end? Or do we add them together and say you have just one I band? We do the first way. We say a sarcomere has two halves of an I band, one on either end. Why? Because that I band was cut in half by the Z disc. And it has one complete A band in the middle. And right in the middle of the A band is an area where you only have your thick filament with no overlap of the thin. We call that the H zone. And in the middle of the H zone is a little protein. We call that line, M because it's in the middle. So this is a lot of proteins and a lot of areas we just happen to name within a sarcomere. And it's these areas and these proteins that are going to be responsible for contracting and getting the muscle and getting the sarcomere, the myofibril, and the muscle to contract. So we're going to have to talk about that. How do you contract? It's really myosin and actin interacting with each other. They pull on each other, kind of the way you would do if you were to interlace your fingers. It's what we call a sliding filament model. Actin and myosin pull and slide along each other. Mainly, myosin pulls actin towards that M line. And when it does, it's going to pull whatever it's attached to. Actin happens to be attached to the Z disc. We could go back and see it in the picture. Again, here's our picture of our sarcomere. And you see the thin actin filament, and yes, it's attached to a Z disc. So when the myosin heads pull actin towards the M line, it will drag the Z discs with it. So during contraction, we're going to see some changes in a sarcomere. You're going to have to know these changes that we see in a sarcomere during a contraction. It all relates to, again, actin and myosin overlapping. When you're resting, everything's nicely spaced out when you're relaxed. But once you contract, again, those myosin heads pull actin towards the M line. So you're going to lose some things. Remember, when you're resting, you have areas where there is no overlap. Remember, those were the I band and the H zone right in the middle. There's no thin filaments mixed in with the thick in that H zone. But when you contract and they slide along each other, you're going to have overlap. So your I band will narrow. You see this pink area has narrowed. And in some cases, it might even disappear. Same thing for the H zone. Remember, that was where there was no overlap. Well, now you do. So your H zone will also narrow and possibly disappear. These are two changes you're going to see. And one more. Look at those Z discs. They're very far apart when you're relaxed. But remember, when you pull actin, you drag the Z disc. So they look like they move closer together. Those are things you're going to see in your sarcomere during a contraction. What are they? I band will narrow and possibly disappear. H zone will narrow and possibly disappear. And the Z discs move closer together. You got to know that. You got to know what you see in a sarcomere during a contraction. We actually got through a lot of this. All right. Now we got to really get into a muscle contraction. You got to know all the steps of a muscle contraction in order and what happens in each step. And to help confuse you a bit, your textbook likes to break the steps down into different phases and it names them. But I've written it all out in this one little document here. Okay, this is part sliding filament model, excitation, contraction, coupling, and the initial excitation phase. Okay, so I'm just going to look at this and then I'll go to the picture. Kind of imagine you're sitting and you want to get up and walk to the door. Well, it first begins with your nervous system. Remember, your nervous system controls a lot of other systems, at least initially. And so if you want to walk, it's going to actually begin with your nervous system. It's going to begin with your neurons. And you remember, your neurons can make action potentials. So it's going to begin with an action potential in a neuron arriving at the end of the neuron. Remember, the end of the neuron is called the axon terminal. And when it arrives at the axon terminal, it triggers the opening of a voltage-gated channel. Oh, before we get into that, it helps to know. Actually, we'll get into that when we get into neurons. I was going to talk about the different types of channels, but we talk about that in chapter over nervous tissue. But for now, let's just go through muscles contraction. So once the action potential arrives at the axon terminal, it will trigger the opening of voltage-gated calcium channels, and calcium is going to rush into the cell. You're going to have an influx of calcium. And that influx of calcium will trigger the release of your neurotransmitter, acetylcholine. And acetylcholine will drift across the synaptic cleft. It will diffuse across the synaptic cleft and bind receptors on the motor end plate. The motor end plate is another name for the plasma membrane of your muscle cell. It is the plasma membrane of your muscle cells, specifically in a neuromuscular junction. Just FYI. Okay. And when acetylcholine binds receptors on the motor end plate, that will trigger the muscle cell to depolarize and make its own action potential. Okay. And this action potential is going to travel along the entire plasma membrane, including those parts of the plasma membrane that dive deep into the cell. Remember, those are the T tubules. Now we're finding out why triad is important. Now we're finding out why you have terminal cisterns on either side of a T tubule. Why? Because as that action potential is traveling down a T tubule, it's going to zap those terminal cisterns. It's going to zap the sarcoplasmic reticulum and cause it to release the calcium that it's been storing. And that calcium is going to travel throughout the cell looking for one thing. It's going to look for and bind to troponin. And when it binds to troponin, it's going to cause troponin and tropomyosin to move away from the areas where myosin needs to bind to actin. We call those myosin binding sites, sometimes called active sites on actin. All right. And calcium only needs to bind to troponin. Think of troponin as the boss. So once you tell the boss what to do, everybody else follows along. So once calcium binds to troponin, troponin and tropomyosin move away. And then at that point, you get to go into your sliding filament mechanism or your cross bridge cycle. All right. Your myosin heads need to bind to actin. And for them to do that, they kind of have to look up at actin. They have to form what's called a cox position. All those little myosin heads need to cock their heads back. And they do it by using ATP. When you use ATP, we say you hydrolyze ATP. Okay. And then once that happens, those myosin heads can bind to actin. And when they bind actin, they form what's called a cross bridge. And then once they form the cross bridge, myosin could pull on actin in what we call the power stroke. It's pulling it towards the end line, towards the middle. And then it's kind of like a game of I forgot the name. It's kind of playing tug of war. There we go. When you play tug of war, you can't just pull once the rope and win. You got to pull and keep pulling. So when myosin heads actin, they need to let go so they could attach and pull some more. So once it pulls during the power stroke, it needs to let go. To do that, it's very simple. It just binds to ATP. And just binding ATP will cause it to release actin. And that ATP it bound itself to is the same ATP it's going to use to form the cocked position. And this will cycle or repeat until you've hit maximum contraction. And that's how you contract. That's how you get up and walk, for example, or raise your hand. Happens extremely fast. But then once you move, you're eventually going to have to relax. To relax, you got to do two things. You got to take all the calcium that you released from the sarcoplasmic reticulum, you got to put it back. And to put it back, you need to use ATP because we use a pump. Active transport. Remember, active transport requires ATP. So you're going to need ATP to put calcium back so that it's no longer binding to troponin. You could go back to kind of protecting and regulating actin and myosin again. And we attack one of the first steps, acetylcholine. You got to break down acetylcholine with an enzyme called acetylcholinesterase. When you do those two things, you can now relax. So that's really how you contract. This chapter spends a lot of time going through those simple steps. All right. But now you know. I'm sorry, I'm just going to picture about skeletal muscle contraction. Sorry. There's a big picture. Actually, before we get to the big picture and summarize it again, this slide helps us with another thing you need to know. You need to know what is a neuromuscular junction, or at least what are the parts to a neuromuscular junction. I put it in your review. It's 3 parts to it. It's an axon terminal, synaptic cleft, and a motor end plate. But I saw the picture, and I can point it all out here. This is a neuromuscular junction. It tells you the name. It's part neuron and part muscle cell. Remember, a junction is connectors between two cells. And we see the three parts here. I see the end to the neuron. It's this yellow stuff. It's the axon terminal. And you even see the little synaptic vesicles containing your neurotransmitter, acetylcholine, inside. But it's just the axon terminal is one. And when we talk about the neuron exciting the muscle cell, talking to the muscle cell, they actually don't need to touch. So there's a little space in between. We call that the synaptic cleft. It's just a little gap of space. And then there's the plasma membrane of the muscle cell. Remember, the plasma membrane, aka sarcolemma, specifically in a neuromuscular junction, is called a motor end plate. So just know those three parts. And you know how to move. But again, I just want to go to a picture and show you how to do the contractions in order. So all these slides are just going through all the steps that involve a muscle contraction. Everything from the beginning of all those different types of channels, but I mentioned we're going to talk about it when we get to nervous tissue and how the muscle cell makes its own action potential as well, and how that leads to the cross-bridge cycle. But there's a summary slide that kind of puts it all together. Oh, did we get there? Back up. Oh, there we go. Almost there. Here it is. This is the big picture slide. Puts it all together in a skeletal muscle contraction. Make it a little bigger. See if it'll let me. No. Okay. But anyways, you can still see what's going on. So again, just stick with the order that's in the review. Very straightforward, very simple. Oh, come on. We lost our slide again. Sorry. Here it is. So to begin, first thing, your action potential arises at the axon terminal, triggering the opening of voltage-gated calcium channels. Calcium rushes into the cell, and that causes the release of acetylcholine. Acetylcholine diffuses across the synaptic cleft and binds receptors on the motor end plate. This triggers the muscle cell to make its own action potential that travels throughout the entire plasma membrane and down a tubule, where it triggers the release of calcium from the sarcoplasmic reticulum. That calcium then binds to troponin, causing troponin and tropomyosin to move away from the myosin binding sites on actin. At the same time, those myosin heads are using, aka hydrolyzing ATP to form the COX position. Then they're binding, forming the cross bridge, then pulling on actin during the power stroke. Then you need to let go by binding a new ATP. And the cycle will repeat until you hit maximum contraction. This one little slide did it all. And you remember the two things you got to do to relax. Put calcium back in the sarcoplasmic reticulum with ATP and get rid of acetylcholine with the enzyme acetylcholine esterase. You know how to contract. And now that you know how to contract, you realize something. It takes energy not only to move, but to relax. It takes ATP to move. You saw it takes ATP to get myosin heads to form the COX position and to let go of actin. And it takes ATP to relax because you need it to put the calcium back into the sarcoplasmic reticulum. This is why muscles need so much energy. They need energy to move and energy to relax. So we got to spend some time talking about energy sources for skeletal muscles, really skeletal muscle cells. You have to know your three major energy sources and how are they giving you energy and for how long. There's three sources. There's something called creatine phosphate. It's a type of what we call an immediate cytosolic source of energy, but it's really just creatine phosphate. Then there's something called glycolytic catabolism, sometimes called glycolysis. And then there's oxidative catabolism, sometimes called aerobic respiration. This is what your mitochondria do. So this is going to make lots of energy. But let's go through. Keep it as simple as I'm about to mention. First up is creatine phosphate. You see that in the picture here. Creatine phosphate is an immediate source of energy that only gives you energy for a matter of really seconds. Okay. How? It's basically a storage container for energy. It's storing it in the bonds. It's a type of chemical energy. How? Let's say you ate breakfast and you're now sitting down and you probably didn't work out and use as much energy's worth of uh, energy as you did consume. So you have energy you need to store. When you need to store it, you're going to use creatine. You're going to take an ATP and you're going to pop off a phosphate and you're going to give it to creatine to form creatine phosphate and it will store the energy. And let's say later on you go to the gym. Now you need that energy. Well, you're just going to take a lesser form of ATP, like ADP, which just has two phosphate groups on it, and you're going to pop off the phosphate group and give it to the ADP to form the ATP, and you get your energy back. That's how it's storing the energy. It's storing it in the bond of the phosphate group. Okay. And so, because of that, you could use up all your creatine phosphate. Once you pop off all the phosphates off your creatine, you've used them up. So, this doesn't give you energy for that long. It only gives you energy for seconds. Okay. So, creatine phosphate kind of stores energy in the form of the phosphate group, and it only gives you energy for a matter of seconds. Your textbook even gives you a number. It gives you 10 seconds. No, you don't need to know 10. Just know seconds. Okay. But we all know, hopefully, we could work out for more than a couple seconds. So we must have more sources of energy. We do. Another one is called glycolysis, sometimes called glycolytic catabolism, sometimes called anaerobic respiration. Okay. But if you call it glycolysis, it gives you a hint. Glycolysis is you breaking down sugar for energy. Lysis means to break. Glyco is for glycogen and glucose. It's you breaking these things down for energy. Okay. That's all it is. You're breaking down glycogen into glucose and then glucose further down for energy. You're doing it without oxygen. That's why it's sometimes called anaerobic respiration. Okay. But there's another. And how long does this give you energy for? Sorry, I didn't mention that. This gives you energy for about a minute. Okay? It doesn't give you that much more energy. It gives you energy, I'll say, for about a minute. Yeah, your textbook says between 30 to 40 seconds, but I don't want you to confuse that seconds with the other seconds of creatine phosphate. So we'll say for about a minute. But again, we could work out, hopefully, for more than a minute. Why? Because we have one more source of energy that gives us energy for hours. It's what we call oxidative catabolism, sometimes called aerobic respiration, because now you're using oxygen. You're still breaking down sugar for energy, still breaking down glucose for energy, but you're doing it with the help of oxygen. So this is aerobic catabolism. And this will give you energy for hours because this results in a lot of energy production. All right. So those are your sources of energy. And how long? Creatine phosphate for a couple seconds, glycolysis for about a minute, and aerobic metabolism or aerobic respiration for hours. And then we get into some global talks about muscle contraction. Like what happens during a twitch? What is a length tension relationship? These are all us just really studying a muscle contraction in a little bit more detail. That's all it is. Okay? So we're going to look at some of this. For example, when we look at a twitch, what is a twitch? A twitch is a very brief singular muscle contraction. And even in a brief singular muscle contraction, we still find out a lot about a muscle cell. Really, when we talk about all those steps of a muscle contraction, it's really from studying very simple contractions like a twitch. And even though a twitch is a very singular contraction, we could still break it down into different phases. When we look at the twitch, we break it down into three phases. There's the latent period or latent phase, the contraction, and relaxation periods. And you see them all in this picture here. The latent period, contraction period, and the relaxation period. If you notice, over time, they don't represent the same blocks of time. The latent period is very brief, okay? Less than 2 milliseconds. Your contraction period is a bit longer, and the relaxation period takes a longer time. It's like your muscles contract. Their contraction behaviors are kind of like a teenager. Oh boy. Like a teenager watching TV. Ask a teenager who's watching Adventure Time to go wash the dishes. They're not going to hear you right away. That's like their latent period. For your muscle cell, this is it waiting. This is you waiting for the neuron to release acetylcholine, for it to diffuse across the synaptic cleft and bind receptors on the motor end plate. That takes time. You need time to get the signal. But once you get the signal, or once your teenager remembers you told them to wash the dishes, they're going to go do it. Your muscles are going to go contract. And once your teenager is done washing the dishes for 2 minutes, they're going to spend 2 years recovering because it was so hard to wash those. Your muscles act the same way. They spend a short period of time contracting and almost half the time relaxing. It takes time to put all that calcium back into the sarcoplasmic reticulum and destroy any remaining acetylcholine using your enzyme acetylcholinesterase. So even though it seems simple, these 3 periods are really those parts of a muscle contraction just broken down. Keep going. There's even a relationship to how long your muscle cell and sarcomere could be and how much of a contraction you could generate. It's called a length-tension relationship. Turns out there's an ideal length you want to your sarcomere. Remember, when you're relaxed, everything's spaced out. So if you have a length-tension relationship where your sarcomere is anywhere between 80 to 120% its maximum length, that's a good area to be to get enough overlap. You need to space things out to give room for myosin and actin to do their sliding filament mechanism, for myosin to pull those actin heads or actin towards the M line. But again, I keep going. A couple more things to know. Like, what is a motor unit? What is a motor unit? What's muscle tension? And there's even different types of muscle cells, skeletal muscle cells. Remember, muscle fiber is just another name for a muscle cell. So we're going to have to look at the different types of muscle cells, mainly type 1 and type 2 muscle cell fibers. A motor unit, muscle tone, and even different types of muscle contractions. And then once we're done talking about skeletal muscle, then we're going to double back real briefly and compare it to smooth and cardiac muscle. So let's look at some of these things. Let's go. This is mentioning the length-tension relationship I mentioned before. If the length of your sarcomere is anywhere between 100, sorry, not 80, anywhere between 100 and 120% of its natural or maximum length, that's a good distance. You spaced out all the myosin heads and the actin to give them enough room to overlap, so you'll get your maximum contraction. If you're outside of this zone, well, you'll see a decrease in contraction strength. That's what you're seeing here. Anywhere between 100 to 120, you will get your maximum contraction, maximum force or tension generated. Anywhere outside that range, less than 100 or greater than 120, you'll see a decrease in strength. There's an ideal length for a contraction to occur. That's all this length-tension relationship is trying to explain. And then, sorry. And then I mentioned we're going to have to compare the different types of muscle cells. There are type 1 fibers and type 2 fibers. Type 1 fibers are sometimes called slow fibers because they contract slower, while type 2 fibers are called fast because they contract faster. And the reason why one is slow and one is fast has to deal with their myosin ATPase activity. Remember, for myosin to interact with actin, it needs to bind to and hydrolyze ATP. For it to hydrolyze, aka use ATP, it needs an enzyme called myosin ATPase. Remember this? Remember ATPases use ATP. Remember there was the sodium potassium ATPase, which used ATP to pump sodium potassium. Well, for myosin to interact with actin and use ATP, it needs myosin ATPase. And how active it is will determine how fast you can move. If you have high ATPase activity, you will move fast. So your type 2 fibers tend to have high myosin ATPase activity. So they burn through the ATP very quickly. And because of that, your type 2 fibers get tired faster. Think sprinting muscles, your leg muscles, those sprinting muscles, or those in your fingers. That's why your fingers can move really fast, but they cramp up and get tired very quickly because it has this high myosin ATPase activity. It's burning through the ATP quickly to get you to move quickly, and you will get tired quickly. It has very weak endurance. Your type 1 fibers, they have low myosin ATPase activity. And because of low myosin ATPase activity, they will burn through the ATP slowly. So the muscle tends to contract a little slower. But because it's burning through the ATP so slowly, you tend to hold a contraction for long times. This has good endurance. Examples of places where you have type 1 fibers are in your back. That's why your back could fight gravity. If your back muscles didn't contract, you would be slumped over all the time because of gravity. But you're able to sit up and stand up. Even if it's not completely straight, like your parents tell you to, you're still contracting. You're doing it for long periods of time, almost all day. And you could do it because your back has a lot of type 1 fibers. So that's the major difference. How much myosin ATPase activity you have will determine how you can move and how long you can hold the contractions. One more time, very simply, type 1 fibers have low myosin ATPase activity, so they burn through the ATP slowly, so muscles contract slowly, but they could sustain the contraction for a long period of time. While your type 2 fibers have high myosin ATPase activity, so they will burn through their ATP quickly to get you to move quickly, but then they will get exhausted quickly as well. So those are your type muscle cell fibers. Keep going. A couple more concepts to know, and we're done with chapter 10. Again, yes, there are other types. We even break down the type 2 fibers, but I'm not going to ask you on that detail of the breakdown of the type 2 fibers. Okay. Going on to another portion of our chapter 10. So now you know what a muscle cell looks like. You know the kind of intimate parts of a muscle cell and how to contract and where you get your energy from, and even the different types of muscle cells that exist. Okay. So now we could get into a little bit more physiology. Oh, sorry. Oh, I think I closed the wrong one. Oh, here we go. Like, what is a motor unit? Remember, for you to move skeletal muscle, it begins with a neuron setting an action potential. So every time you move your skeletal muscle, it's technically thanks to the nervous system. So your nervous system will even impact how great of attention, not only just moving, but how great of attention you could produce. Meaning, can I lift up a 5-pound dumbbell with a light amount of tension, or a 20-pound dumbbell with a lot more tension? So we got to discuss a motor unit. A motor unit is simply a motor neuron, which are the neurons responsible for exciting, and all the muscle cells will talk to. To innervate means to excite or talk to. That's all this is. And so if I want, let's say, a very light contraction, I only want to pick up something that weighs 1 pound. I will only need a few motor units because I don't need to excite that many cells because I only need to generate enough force to lift a pound. But let's say I want to lift up a 20-pound dumbbell. Well, I'm going to need to excite more cells. So I need more motor units to activate more muscle cells to get me to lift a larger structure, to generate a greater force to lift a larger structure. We call this recruitment. Recruitment is literally that. You recruit more motor units to generate a bigger tension. Why? Because you're activating more of the muscle cell. That's a little motor unit. And now I get to explain something. Oh boy, I lied to you. Oh boy, how did I lie? Turns out I lied when I told you you control skeletal muscle tissue, at least a little bit. Remember, skeletal muscle tissue is voluntary, but not all the time, not everywhere. There are some cases where you have involuntary contractions of skeletal muscle tissue. We call that muscle tone. And you need this. There's good reason to have involuntary contractions of skeletal muscle. For one, it helps you to produce heat. You know this way back from chapter 1. You know this way back from when we taught negative feedback. Remember, when you get cold, you're going to warm up. How? By shivering. Shivering is your body moving. It's moving because of skeletal muscle. But you don't do the shiver. When you get cold, you don't say, excuse me, I got to go shiver for a second. No, your body does it involuntarily. So sometimes you do have skeletal muscle moving involuntarily. We call that muscle tone. It's good for things like heat production, things shivering. It's good for things like your posture, your back. Remember, I told you you're fighting gravity constantly with your back muscles, but you're not thinking about it. You're not saying, man, I'm so tired of fighting gravity today. You don't think about it because that's muscle tone you're experiencing. You're experiencing your skeletal muscles contracting involuntarily. That's all muscle tone is. And there's good reasons for it. We look at the different types of contractions. Let's go look at it. Not only do you know that you can contract, now you know there are different types of contractions. We group them up. There are two major types. There is an isotonic contraction and an isometric. You got to know the difference. And like always, their names give you a hint. Just break down the names. Remember, iso means the same. Tonic in this means tone or muscle tension. So when you say isotonic, you're saying this tension stays the same. So what's going to change? It's going to be the length of the muscle. This is what people normally do when they lift weights. When you lift weights, the tension stays the same, but the muscle length will vary. How? Let's say you pick up a 20-pound dumbbell. And when you contract and you bring the dumbbell up, well, you still got to generate 20 pounds. And when you bring the dumbbell down again, put your arm back down, it's still 20 pounds, no matter what. When you're contracting or relaxing in that dumbbell bicep curl, you still have to generate 20 pounds of tension to hold that dumbbell. But when I bring the dumbbell up, my muscle shortens. And when I bring the dumbbell down, my muscle lengthens. You're experiencing an isotonic contraction. Again, the tension, the force will stay the same. Here's the picture. No matter where you put the dumbbell, you need to generate 20 pounds of force to hold it. But sometimes during the contraction, the muscle will be shorter, sometimes it will be longer. The length might change. That's an isotonic contraction. And we even break it down. We break it down into those two instances of when the muscle length changes. We give a name to when the muscle length is short and when the muscle length is long. When the muscle length is short, we say you're in the concentric phase of your isotonic contraction. That's what you see up here. It's in the concentric phase because the muscle length is short. And when the muscle length is long, we say you're in the eccentric phase or you're in the isotonic eccentric contraction. That's all that means. Okay, short muscle, concentric, long muscle, eccentric. And the muscle is changing, muscle length is changing because this is an isotonic contraction. Only the tension stays the same. But then there's the opposite type of contractions. It's an isometric. Again, iso means the same, metric as in unit of measurement. The metric system. So in this case, now the muscle length stay the same, but now the tension or the force you're generating will change. This is something like pushing on a wall. You could push on a wall very gently, giving a little bit of force, or you could push on the wall very hard with a lot of force. But no matter what, you're pushing on the wall. Your muscle length doesn't change. You're not doing a push-up on the wall. You're pushing it. Another example is holding some books in your... I'm old. I don't know if you guys know this. I'm an old lady. When I was in school, they used to punish us by having us hold books in our hands all day. Well, when you're holding the book, well, your muscle length is not going to change. You got to keep your arms right out in front of you. But your teacher could be mean and add more books while you're standing there. So you're going to have to change the tension or the force generated. Oh, and your teacher was punishing you, Dr. Higgs. You were experiencing isometric contractions. Oh, boy. Okay. And I even know your different types of contractions. Keep going. And we even talk about training. We talk about exercising and the effects exercising have on your body. Okay. Talk about exercising. We talk about training. There's two types. There's endurance training and there's resistance training. And you're going to have to know the changes you're going to see in both of those trainings. Yeah. And again, you might know this from your everyday life. Remember, in anatomy, it's just that we give fancy names to stuff. For example, you probably know what endurance training is. Think running. Yeah. When you go running, you're doing a type of endurance training. Okay? So think of what changes you might see if someone were to start running every day, all day, to probably get ready for a marathon. Well, what are you going to see? For one, you're going to see increased fatigue resistance. This is endurance. So you're not going to get tired as fast. Okay? Kind of thing. At the start of your marathon training, you couldn't run 5 miles because you got tired too quickly. But by the end of your marathon training, you should be ready to run a 10, 20-mile marathon. Why? Because you're not tired as fast. Why? Because you have increased fatigue resistance. That is a result of endurance training. And endurance training focuses on repetition. That's why I'm saying running. Running is just you repeatedly moving your legs. When you hear the word repetition with light weights, think possibly running. That's a type of endurance training. Keep going. And if you're wondering, why do you have increased fatigue resistance? Well, for that, you got to think about your sources of energy. Remember, you get energy from things like mitochondria. Reason why you're increased fatigue resistance is because you're having increased activity of enzymes in the mitochondria. Think those myosin ATPases. Their activity is increasing, so you have energy to fuel your contractions. And not only do you have increased enzyme activity, you get better at using your sources of energy. Things like fatty acids and non-glucose sources, your body gets more efficient, meaning they get better at using them energy. Yeah. This is why, again, that runner could go eat a pizza, same amount of slices as you, and seem not to gain as much weight. If I look at a pizza, I'm going to gain 10 pounds. Why? Because I don't do endurance training. Okay. And what else? You even have an increase in blood vessel networking, meaning you literally develop blood vessels to help connect different parts of the body. Your body's getting more efficient at supplying energy when you do endurance training. These are effects you're going to see. Okay. And then there's strength training or resistance training. Okay. Think lifting weights. Strength training, aka resistance training, that's lifting weights. So you're going to see heavier loads with fewer repetitions. Think when you watch The Rock on Instagram work out at his home, he does like 2 bicep curls and he drops the weights and walks away. It's fewer repetitions because you're focusing on a heavier load. That's the goal of resistance training. So think, what happens to a person when they start lifting weights? Think of bodybuilders. Oh, what happens with? Turns out they have an increase of motor unit activation. Meaning they're going to get more of those neurons to talk to more of the muscle cells, so they activate more of their muscle because they need to generate a larger force because they have a heavier load. What else? They even have an increase in force production. Again, this is all because of increases in motor unit activation. If you can activate more of your muscle cell, yes, you're going to generate a greater force to lift up that heavier weight. What else? We'll see something called hypertrophy. Hypertrophy is when a cell gets bigger. And we see that. You know, when someone starts lifting weights and becomes a bodybuilder, they get bigger. And the reason why is not because they get more cells. Believe it or not, The Rock has just as many muscle cells as I do, even though I don't look like I have a single muscle in my body. Me and The Rock have the same number of muscle cells. It's just that the rock has bigger muscle cells because they've done hypertrophy because of his resistance training. So what are we going to see? We're going to see better coordination and more coordination between neurons and muscle cells. And we're going to see the muscle cells get bigger. They're going to hypertrophy. But I don't know if you've ever seen a bodybuilder go running. Oh, if you've ever seen a bodybuilder go running, it's both hilarious and sad at the same time. Those guys can't run to save their lives. Why? Because they actually have decreased endurance. That happens with resistance training. Remember, you're doing less repetition, so you're not helping your endurance. And they'll have a decrease in all those mitochondrial proteins as well. Yeah. And because of this decrease, that's why they're having the decrease in endurance. Remember, you need that higher activity for the endurance. So those are some differences in body composition and physiology that we'll see in the different types of training. And muscle fatigue is just you running out of energy. So when you run out of energy, you get tired. That's muscle fatigue. It's you literally depleting your sources of energy. And then to finish off, we kind of do a slight comparison of skeletal muscle tissue to cardiac and smooth muscle tissue. And again, you know the differences. We're looking inside the cells, yes, but before we look inside the cell, we know some things if we remember these tissues. Okay. There are going to be some similarities between smooth, cardiac, and skeletal, and some dissimilarities. For one, you know that cardiac and skeletal muscle tissue are striated. And you now know a striation is just alternating light and dark bands. It's alternating A and I bands. So you know because the cardiac muscle tissue has striations, you know it has sarcomeres with A and I bands in it. Oh, right off the bat. What else? We know smooth muscle tissue, like cardiac, typically has just one nucleus per cell. So we're going to see that when we look inside the cell. We know smooth muscle tissue is non-striated. So what does that mean? It means it's missing those alternating light and dark bands. It does not have A and I bands. Just by kind of thinking about the tissues and what they look like under the microscope, you already know some differences between these muscle tissues. And you know what they do. You know they all help with movement. It's just a matter of where. And you know where to find them. For example, you know to find smooth muscle tissue in hollow organs like your intestines. So it's going to help to move and regulate the flow through your hollow organs, like your intestines and your GI tract, that's your gastrointestinal tract, or in your blood vessels. Your blood vessels are hollow organs, and yes, they have smooth muscle in them to help move your blood along. That's the job of smooth muscle tissue. It's still helping with movement, it's just a matter of where. And because it's doing movement, we know it's contracting. And we know in order for it to contract, it must have contractile organelles. It does. It does have the actin and myosin filaments. And actin filaments are still attached to a structural protein. It's just not the Z disc. Remember, there are no Z discs here because there are no A and I bands. But it does have a structural protein very similar. It's called a dense body. Dense body in smooth muscle is akin to Z discs in skeletal muscle tissue. So it's still providing an anchor, a structural support for your actin filament. And again, it still has myosin filament as well to do the interactions because it does contract, it does move. But there's something else. Turns out smooth muscle doesn't have as big a sarcoplasmic reticulum. And because it does not have as big a sarcoplasmic reticulum, it actually does not have T tubules. Okay. So that's a little bit on some differences between smooth and skeletal. I mentioned it doesn't have that much of a sarcoplasmic reticulum. So if it doesn't have that much of a sarcoplasmic reticulum, where is it going to get its calcium from? If it only has a little sarcoplasmic reticulum, it still needs calcium to do the contraction. So where is it going to get it from? That's what we talk about here. Turns out it's going to get its calcium from outside the cell. We call it extracellular calcium. That's calcium outside of the cell. So during a contraction in smooth muscle tissue, because you do not control it, we do not begin with an action potential arriving at the axon terminal. It all happens in the smooth muscle cell itself, starting off with an influx of this extracellular calcium. Calcium rushes into the cell from the outside. And when it rushes into cell, it still binds to a regulatory protein. It's just not troponin. It's called calmodulin. And when it binds to calmodulin will then activate another protein called myosin light chain kinase, or MLCK for short. And then MLCK, or myosin light chain kinase, will activate, your myosin ATPase, and you could begin your cross-bridge cycle of myosin hydrolyzing ATP to form the cocked position, binding to actin to form the cross-bridge, and pulling during the power stroke. The rest is the same, but this initial part is different. This is why you don't control smooth muscle. You don't see any nervous system here. If you had to break it down, what organ am I? Think you are your brain. And you don't see any nervous system structures here. This is why you don't control smooth muscle. The differences between smooth muscle and skeletal muscle tissue. Okay. Don't worry about the latch phase. And then we got to look at cardiac muscle tissue. Again, lots of similarity to skeletal. Just like skeletal, it has intercalated discs, so it has sarcomeres with alternating A and I bands. So you could establish all the same anatomy for your sarcomere that you saw in the skeletal muscle tissue in your cardiac muscle tissue. What else? Remember, we saw intercalated disc in cardiac. And I told you those intercalated discs were part gap junction, allowing cells to send information, their electricity, from one cell to another. Remember, this is how you get the heart to contract as one. You call it a coordinated unit. It contracts as one. Why? It's thanks to all those gap junctions in the intercalated discs. Remember, you don't control cardiac muscle tissue either. So how are they contracting? Well, it's because of the cells. There are two major groups of cells in your cardiac muscle tissue. They're your pacemaker cells and your regular contractile cells. And it's the pacemaker cells that's going to help with the initiation. Remember, in skeletal muscle tissue, it began with an action potential arriving at an axon terminal. Turns out, your pacemaker cells in your cardiac muscle tissue are the cells generating the action potential. And they could spontaneously generate it, meaning they could generate an action potential out of nowhere. Okay. And once it generates the action potential, it will spread it to all the contractile cells around it via those intercalated discs so that you could get a coordinated contraction. So these pacemaker cells and their autorhythmicity, their ability to generate their own automatic rhythm, as to why you cannot control cardiac muscle tissue. All right. So that's it really for chapter 10. And then there's chapter 11. In chapter 11, it's now the nervous system and nervous tissue's turn. And again, you know some things already, way back from chapter 1 when we did all the organ systems. Remember, nervous system has 3 organs: brain, spinal cord, and nerve. And those brain, spinal cord, and nerves are helping us to do a couple things. For one, you know now your nervous system helps you to move. So one function of nervous system is to help with movement, really by stimulating your muscle cells. Think back to chapter 1. Remember I told you it also helps with sensation. Okay. So another function of the nervous system is helping you to sense things in the body. What else? Remember, your nervous system can even help to process information. Your brain is like a computer. You could process information. Your nervous system does a lot. Okay, and a lot of that functionality of the nervous system overlaps with other organ systems, like movement. That's a function of skeletal and muscular system, but we now know it's also a function of nervous system. Okay. And I mentioned the organs. It's only 3. It's brain, spinal cord, and nerves. And you might recall way back that we break up those 3 organs into 2 groups. We break up the nervous system into 2 major systems. There's the central nervous system and the peripheral nervous system. Okay. Remember, your central nervous system is the brain and spinal cord thing. They're in the center of the body, so they're the central nervous system. While all your nerves are peripheral nervous system. You got a lot of them. All these yellow creepy lines in the sky are nerves. Some of them come off the brain, so we call them cranial nerves, and some come off the spinal cord, so we call them spinal nerves. All right. That's what you need to know in your exam review. Know the major divisions of the functions of the nervous system and their major divisions, and what organs are found in each. Central nervous system, brain and spinal cord, peripheral nervous system, cranial nerves, and spinal nerves. And when we look at all those nerves in the peripheral nervous system, they do a lot of work. So we got to break down the functions of the peripheral nervous system. Again, going back to your slide. This little picture breaks down the peripheral nervous system functionally. Here it is. And think of those major functions I mentioned. It can help you to feel and it can help you to move. So when we look at the peripheral nervous system, we break it up into a sensory division and a motor division. They help tell you the name. The sensory division helps you to feel. But you can feel in different places. So when we look at the sensory division, we break it down even further into a somatic sensory division and a visceral sensory division. Think of where you need to feel stuff. Do you need to feel stuff like on your skin or your muscles, skeletal muscles? Well, that's your somatic sensory division. So if you feel anything on your skin, you're feeling it with the somatic. But you could also feel things inside your body, your internal organs. That's a visceral sensory division. Okay, think when you have a tummy ache because you ate all those cookies by yourself. Well, you're experiencing that pain thanks to the visceral sensory division. And then there's the movement. There's a motor division. Again, think you got different muscle tissues. Some you control and some you don't. When it comes to skeletal muscle tissue, the voluntary one, that's somatic motor. But when it comes to the involuntary ones, that's cardiac muscle tissue and smooth muscle tissue, and also glands, well, that's autonomic. You don't control this system. And you don't control these tissues in this system. So those are the functional breakdowns of your peripheral nervous system. And then we go and we look at the tissue again. And we already know the two cells found in the tissue. It's the big neurons and it's the little neuroglial cells. Remember, we did this in the lab already. You're just revisiting nervous tissue again. Remember, when we looked at a neuron, we could break it up into different parts. Remember the big central globular part? That's the cell body. And then you have extensions coming off the neuron. Remember, the long extension is called the axon, and the short extensions are called the dendrites. Remember, the dendrites are how you receive information, and the axon is how it sends out information. You remember all the little round cells are the neuroglial cells, helping to support the neuron. We knew that. We knew that already. Here's another picture of our neuron to help break down some more parts. Again, here's the cell body, sometimes called the soma, S-O-M-A. Contains most of the organelles like the nucleus. Then you have the long extension, the axon, ending at your axon terminals. And when you look at the axon, it even has branches. We call branches of an axon collaterals. What else? Look at the axon. You see this blue thing sitting on this yellow neuron in this cartoon. These blue things are fat. We call them myelin sheath. It will play a role in helping the neuron to its action potential. What else? And if you look at the myelin sheath, you notice it's not covering up the entire axon. There are spaces where the axon is exposed. We call those nodes of Ranvier. They're just where your axon is missing the myelin sheath. Where it has myelin is called an internode, but where there is no myelin is called a node of Ranvier. And again, we see the short branches are our dendrites, helping to receive information. What else? We even named the beginning to an axon. The beginning is called the axon hillock. That's just the beginning to the axon. And your axon is still part cell, so it still has the plasma membrane holding in the cytoplasm. It's just, again, names change. The plasma membrane, specifically on the axon, is called the axolemma. And the cytoplasm inside of the axon is called the apoplasm. All right. So those are some parts to your neuron. And one more, it has organelles in it, like a nucleus and an endoplasmic reticulum. But guess what? The endoplasmic reticulum in your neuron also has a different name. It's called a nasal body. All right. It's really the rough endoplasmic reticulum. So if I were to ask, what does a nasal body do? You know already. You know what a rough endoplasmic reticulum does. It helps to make protein. All right. So those are parts to a neuron. And then you got to look at a neuroglia. Actually, you got to look at neurons up close. Turns out you got lots of neurons. And like always, when there's a lot of something, we got to classify them. And when we look at the neuron in our body, well, we could classify them structurally and functionally as well. So we got to do that. We got to look at the three structural classes and the three functional classes of your neurons. Let's go look at them. First up are the three structural classes. There are multipolar neurons, bipolar neurons, and pseudo-unipolar neurons. If you use another textbook, they sometimes call the pseudo-unipolar neurons unipolar neurons, just in case you use a different textbook. And then there's the functional classes, also three of them. It's related to some of the functions of the nervous system. Number one function is to sense things. So there are sensory neurons helping to do that. Remember, another function is to help you to move. That's the motor neurons helping to do that. And I told you could even process information. There are interneurons or association neurons doing that. So you got to know your three structural and functional classes and how are they related. Turns out the majority of your neurons are multipolar neurons. Why? It's because your multipolar neurons structurally are also motor neurons and interneurons. You see that? It's here. Multipolar neurons are your interneurons, and multipolar neurons are the motor neurons. But why are they called multipolar neurons? What do they look like? Because that's a structural name. So it's describing what it looks like. A multipolar neuron structurally has one axon and multiple dendrites. So when you look at this picture, you're actually looking at an example of a multipolar neuron. It has one axon and multiple dendrites. Turns out they do a lot of the work of the nervous system. They are your motor neurons and your association or your interneurons. While your bipolar and your pseudo-unipolar neurons are your sensory neurons. It's just that they help you to sense different things. Your bipolar neurons help you to sense things like smell and vision, while your pseudo-unipolar neurons are helping with some somatic sensory things like pain and touch. All right. But again, we're talking about structurally. What do they look like? Bipolar neurons also have one axon, but they also have one dendrite. And in pseudo-unipolars, it's weird. The axons and dendrites have fused together into one long process, one long extension. That's what you're seeing in these pictures here. So multipolar neurons all have one axon, multiple dendrites. Bipolar neurons have one axon and one dendrite, and pseudo-unipolar neurons, the axon and dendrites have fused together into one long process. Your multipolar neurons are the motor neurons and interneurons. Your bipolar neurons are a sensory neuron helping with things like vision and smelling, while your pseudo-unipolar neurons are also sensory with some general somatic sensations like pain and touch. That's your classifications of your neurons. Then we got to look at your neuroglial cells. Remember, they were the little round dots we saw under the microscope. But those little round dots all have specific functions. There are actually several different cells. There are 6 different cells. You got to know your 6 neuroglial cells. Where do you find them? And by where, it's either central nervous system or peripheral. And what do they do? So this one slide gives us 2 of the 3 things we need to know. It names them and it tells us where to find them, so that all we need to do last is figure out what they do. So let's go through them. There are astrocytes, oligodendrocytes, microglial cells, appended cells, Schwann cells, and satellite cells. And they find in different places. These first four, astrocytes, oligodendrocytes, or microglia, and ependymal cells, are all found in the central nervous system. So we'll find them in the brain and spinal cord. While your Schwann cells and your satellite cells are found in the peripheral nervous system. So we'll find them in your nerves. So let's see what they do. Starting off with the astrocyte. The astrocyte is this green cell you see in the picture. Seems to be touching this neuron and this blood vessel, almost like it's separating them. It's because it is. Because one function of an astrocyte is to form a blood-brain barrier. It literally tells you in the name. It's a barrier between your blood and blood vessels and your brain and nervous tissue. Why? Why do we need this barrier between our blood and our nervous tissue? Because these are cells and they need oxygen and nutrients and they get them from the blood like everyone else. But you need to protect them at the same time. Because there are some things that could get into blood that you don't want in your nervous tissue, called things like infections and toxins. You get infections in the blood. It's called septicemia. You don't want that to spread to your brain and spinal cord. Hopefully it won't because you're protected by this blood-brain barrier created by your astrocytes. Or toxins. Certain toxins get into the blood that, again, you don't want getting to the brain and spinal cord. Again, hopefully it doesn't happen because, again, you're protected by the astrocytes and the blood-brain barrier that they form. That's a major function. Astrocytes form a blood barrier as protection. Keep going. Another cell is called the oligodendrocyte. So these blue cells you see sitting on the neuron. Looks like a blue cell and looks like it's wrapping a part of itself around the axon. It's because it is. It's literally stretching out its plasma membrane and wrapping it around the axon. Remember, your plasma membrane is mainly fat. Oh, remember, I gave a name to the fat sitting on the axon of your neuron. Remember, we call that myelin, or the myelin sheath. That myelin needs to be made, and it's made by your oligodendrocytes. Oligodendrocytes make myelin in the central nervous system. All right. And it turns out an oligodendrocyte can make myelin from more than one axon. Yeah. Keep going. Not the only cell. There are microglial cells, or microglia. They're red cells in the picture. These are basically white blood cells. Remember, you see white blood cells all over the place. You saw it in your skin as your dendritic or your Langerhans cells. You even have it in your nervous tissue. And what do white blood cells do? They fight. They fight and prevent diseases. How? They literally eat them. Remember, phagocytosis is cell eating. So a phagocytic cell is a cell that eats things. So if a microglial cell sees something that does not belong, it's going to eat it in order to protect you. Yeah. And then there's the ependymal cell. These cells tend to line cavities in the brain and spinal cord, and it makes cerebrospinal fluid. That's it. It lines pockets or hollowed cavities in the brain and spinal cord, and it makes cerebrospinal fluid. And this fluid will provide some protection. So those are the cells of the central nervous system. Keep it simple. This one slide summarizes all those central nervous system cells. And then there's the peripheral nervous system. Remember, there's only two, Schwann cells and satellite cells. Schwann cells are a lot like oligodendrocytes, but just for the peripheral nervous system. How? Just like oligodendrocytes, Schwann cells also make myelin for axons. It also stretches out its plasma membrane and wraps it around an axon. Yeah. Process of making myelin is called myelination. So your Schwann cells, like your oligodendrocytes, do myelination. It's just that a Schwann cell could only make myelin for one axon, where an oligodendrocyte can make myelin for multiple axons. And then there's the satellite cell. It's called the satellite cell because when you look at it, it seems to kind of look like it's orbiting the cell body, kind of like how satellites orbit the planet. So we call them satellite cells. But your satellite cells just provide support. To be honest, at this point, we're not quite sure, scientists aren't quite sure how it's helping. They just know that it does. How? Well, when you take away the satellite cells, the neuron suffers. And when you give it back, the neuron does okay. So we know it helps somehow. We're just not sure how. So if I were to ask on an exam, what does a satellite cell do? The answer is it provides support. And if I ask how, well, say I don't know, because we don't know. Okay. If you find out, I guarantee you will get a Nobel Prize. And that's it for your peripheral nervous system. That's it for your neuroglial cells. Okay. And then the rest of this gets into the physiology of action potential. Okay, how do you generate an action potential? How do you propagate an action potential? And all the things related to an action potential. And when you generate an action potential, it's all dealing with the plasma membrane. So we got to look at the plasma membrane. Remember, your plasma membrane is made up of a phospholipid bilayer where you have those hydrophilic heads facing the water either outside or inside the cell, and all those hydrophobic tails kind of clustered and sequestered kind of centrally inside of the plasma membrane. What you might not know or recall is that there are also charges along the plasma membrane. And we even know the charges. We know your plasma membrane has a slightly negative charge on the inside and a more positive charge on the outside. We give a name to this. We give a name to this difference in charge across the plasma membrane when your cell is at rest. It's called your resting membrane potential. Okay. Membrane potential is just the difference in charge across the membrane, and it's when the cell is at rest. And we know that on the inside portion, you tend to be negative, and on the outside portion, you tend to be positive. This is when your cell is resting. And we can measure this difference in charge across the membrane. We can measure your resting membrane potential. This is electricity. So when we measure it, we use the same units of measurement we would use for electricity. We use voltage. All right. Voltage is just us measuring the difference in charges across that membrane. But you got to recall, this is a cell. We're dealing with a neuron. And so these difference in charges across the membrane are going to be really small. So we don't use volts, we use millivolts because it's really small change. And we measure, we know the average resting membrane potential for a neuron. Got to know this number. It's negative 70 millivolts. What does that number mean? It means the cell is 70 times more negative on the inside than it is on the outside. That's all that number means. Okay. We're going to begin to talk about an action potential. And it all begins with knowing when your cell is at rest, it's 70 times more negative on the inside than it is on the outside. You have a resting membrane potential of negative 70 millivolts. And what causes that? Oh, you might recall your plasma membrane has some integral proteins that act as channels to allow things to pass in and out of the cell. And that's actually going to help to establish this resting membrane potential. And we know which one of those integral proteins does it. It's called the sodium potassium pump. So the full name is the sodium potassium ATPase. Remember, your sodium potassium ATPase kicks out 3 sodiums for every 2 potassiums it lets into the cell. So how do you get negative on the inside then? Because if you look, sodium and potassium both have a positive charge. So where did the negative charge come from? It's a numbers game. Okay, think about the numbers game. I told you kick out 3 sodiums for every 2 potassiums you let in. You're kicking out more positive stuff than you're letting in. So that over time you become negative on the inside and positive on the outside. Now your sodium potassium ATPase is establishing your resting membrane potential. It's kicking out too much positive stuff, so you're positive on the inside, and letting in too little positive stuff to the point where you're negative on the inside. So that's your resting membrane potential. But we're going to have to generate a spark of electricity. We're going to have to generate an action potential. And to do that, we're going to have to change this charge difference across the membrane. And in order to change that difference in charge across the membrane, we're going to need some other integral proteins. We're going to need some protein channels. So now we got to talk about some of the different channels we could see inside of your plasma membrane. All right. Some channels are always open. We call them leaky channels, kind of leaky faucets. It's always running. Okay, these are always letting things in and out of the cell. An example is your sodium potassium ATPase. It is always kicking out sodium and letting in potassium. But some channels are closed and you need to do something to open them. We call them gated channels. It's like they have a gate and you need to unlock the gate before you open it. Okay. And we look at different gated channels up close. Okay. If you're a ligand gated channel, this is a channel that's normally closed and you need a specific chemical to open it. The specific chemical is called a ligand. Some channels are typically closed and you need to change the voltage across the membrane to open. You need to change that charge across the membrane to open it. We call those voltage gated channels. And some channels, you need to provide some type of mechanical manipulation. Remember, when you hear the word mechanical, think physical. So think touching it, pressing on it, shaking it, stretching it. You need to do some type of mechanical stimulus to open this channel. So we call it a mechanically gated channel. And so these channels, particularly your voltage gated channels, are the ones that's going to help us to generate the difference in charges across the membrane and to change it for your action potential. Remember, I mentioned the sodium potassium ATPase is a leaky channel. It's always kicking out 3 sodiums to let in 2 potassiums. So it's a numbers game. So over time, you have more positive stuff on the outside. That's how you get your resting membrane potential. But to generate your action potential, you're going to have to change the difference. For one, you're going to have to change it from being negative on the inside to positive on the inside. We call that depolarization. Depolarization is when you become more positive on the inside, or less negative is another way of saying it. But then as soon as you depolarize, you actually got to go back to being negative again. You got to go back to having these poles on either side of the membrane, where you're positive on one end and negative on another end. When you go back to having this polarity, say you've repolarized. To repolarize is you just going back to being negative on the inside again. But your neurons, it's like they do too much. They become more negative than they were at their resting membrane potential, than they were to start off with. They hyperpolarize. These are some of the steps involved in the generation of an action potential. And you're going to have to be able to interpret a graph of an action potential. So I'm going to go through the steps of an action potential using this graph. And these pictures here are us generating the graph that I'm mentioning. Let's go through the steps in the generation of an action potential. How do I make this little spark of electricity? Well, for one, I need to open some voltage-gated sodium channels. In order to do that, I need to become a little bit positive on the inside. And we know the number I need to hit. You need to go from negative 70 millivolts to negative 55. When you get to negative 55 millivolts, we say you've hit threshold. That's the first thing you need to do in the generation of your action potential. You need to hit threshold. You have to have a local potential where you become slightly positive, enough to bump you up to negative 55 millivolts. Why? Why do we need to do this first step? Well, it has to deal with the next step. It has to deal with depolarization. That's step 2. In depolarization, you open voltage-gated sodium channels. Allow sodium to rush into the cell with their positive charges, and that will make the cell positive. How did you open this voltage-gated channel? You changed the voltage. Literally, you went from negative C to negative 55. That's why you need threshold. You need threshold to open your voltage-gated sodium channels. And once you've opened your voltage-gated sodium channels, sodium rushes into the cell, bringing its positive charges with it, and the cell becomes positive on the inside. We call that depolarization. And you become positive all the way up to about positive 30 millivolts. Yeah. And that pretty much makes the action potential. That's the spark. But we're not done yet. You need to reset. You got to go back to normal. Remember, you got to repolarize. In order for us to repolarize, for us to go back to being negative on the inside, well, for one, you got to stop letting positive things in. So you got to close your sodium channels. But that's not enough to get you to go back to being negative. Oh, you got to kick some positive stuff out. Now you're going to open voltage gated potassium channels and you're going to kick out potassium. And that potassium is going to take its positive charges with it, and the cell's going to go back to being negative on the inside. That's repolarization. But I told you, they kick out too much potassium and become more negative than they were to begin with. They hyperpolarize. But no worries, you'll close those voltage-gated potassium channels, and your sodium-potassium ATPase, which is always open, will get you back to your negative 70 millivolts. That's all your steps of a action potential. And it's all in that one little chart. Here it is here. And you start off at rest, where your sodium potassium ATPase keeps you at negative 70 millivolts. Then you do a little local potential, changing the potential across the membrane to about negative 55 millivolts. Remember, that's called hitting threshold. That will open up voltage gated sodium channels, and sodium will rush into the cell, causing you to become positive. That's depolarization. At that point, you've made your action potential. So now you could close your sodium channels, open potassium channels, and kick out potassium and become negative again. Remember, that's repolarization. But oops, you kick out too much potassium and you become more negative than you were to begin with. You hyperpolarize. At that point, you close your potassium channels, and your sodium potassium ATPase, which is always open, will get you back to your negative 70 millivolts. That's your action potential. All right. Those are steps in the generation. Oops, sorry. Those are the steps to the generation of an action potential. Hit threshold, then depolarize, repolarize, and then hyperpolarize. And we could all see it on that chart. Now you know the steps, we could talk about some refractory periods. There are periods of time where you do not send out an action potential. We call it refractory period. Why? You need time to reset. You need time to go back to being negative on the inside again. We call these your refractory periods. You have an absolute refractory period and a relative. The absolute refractory period is when you absolutely cannot generate another action potential. Why? You're in the process of doing it. You see this in the graph. Your absolute refractory period does involve depolarization. So if you're in the immediate process of making an action potential, obviously you cannot make another one. You're too busy. But if you give the neuron a little time, let it get into some repolarization, if you were to give it a big enough stimulus, it can generate another action potential. Why? It's because now you're in the relative refractory period. In the relative refractory period, you can generate an action potential if you have a big enough stimulus. Because you see, you've gotten enough negative again. So those are refractory periods. And it makes sense when you think about it. It's going to sound horrible, but it kind of makes sense. Imagine if you were to prick your finger with a pin. You would feel it. Ouch. Okay. And you're feeling it because the neurons in your fingers sent that sensory information to neurons in your nerves, to neurons in your spinal cord, and neurons in your brain to let you feel that pain. But imagine this. It's going to sound horrible. Don't judge me. Don't judge me. Imagine someone stabbed you in the hand. Uh-oh. Oh, no. Horrible. Dr. Hayes, get your life. Okay. Someone stabs you in the hand. And as soon as someone stabs you in the hand, someone pricks you with a pin right next to it. Do you think you're going to feel that pin prick right next to that stab wound? Probably not, because your neurons are too busy telling you the pain of the stab wound. They're too busy generating an action potential. They're in their absolute refractory period. But if I waited long enough, I'd be able to feel the pin prick once my neurons get into the relative refractory period. There's a reason for these periods. You need to give neurons time to recover. So that's a little bit of absolute and relative refractory periods. You know how to make an action potential. And now that we know how to make an action potential, now we got to move it. Okay, we got to propagate it. We're talking about electricity. Moving electricity is propagating or conducting electricity. So how do we propagate or conduct an action potential? How do we move it along the axon to get it all the way to the axon? Well, like always in your body, you have options. you have two types of propagation. There's saltatory conduction and there's continuous conduction. And there are both ways we're going to move our electricity along our action potential. So what's the difference? Let's do continuous conduction first. And it kind of tells you in the name. Continuous conduction is where you continuously depolarize, repolarize, and hyperpolarize every single part of the axon. Okay, that's how you're going to move it along the axon. You got to go through all of those steps. Depolarize. Then once one area is done depolarizing, it will have to repolarize and hyperpolarize. Do those steps for every single part of the axon. You cannot skip anything in continuous conduction. But that's going to take a while. If you have to imagine doing all those steps in every single part of the axon, that will slow you down a bit. So there's a faster type of conduction. It's called saltatory conduction. In saltatory conduction, you do not need to do those steps in every single part of the axon because you skip some parts. You get to skip the parts that have myelin. Oh, remember we named the parts that have and do not have myelin. Remember the parts that have myelin are called internodes, and the parts that do not are called nodes of Ranvier. In saltatory conduction, you only need to do those steps of depolarization, repolarization, and hyperpolarization in the nodes of Ranvier. So we say the action potential in saltatory conduction jumps from node of Ranvier to node of Ranvier. So you can imagine this will get to the end faster. Kind of imagine having to drive to school. If you did continuous conduction driving to school, well, then you would have to drive all the road to get to the school, which is what we normally do. You can't skip any of the road. You got to drive the whole way. But if you were to do saltatory conduction on your drive to school, that will be like you and your car jumping from intersection to intersection. You don't have to drive the whole road. So you could imagine you will get to school faster if all you had to do was jump from intersection to intersection. This is why saltatory conduction is faster, because you only jump from node of Ranvier to node of Ranvier. So you'll get to the axon terminal faster. And we even know some differences. The reason why you can't jump from node of Ranvier to node of Ranvier in continuous conduction, you can see it in the picture, is because there is no myelin. So there's no internode or node of Ranvier. So we know saltatory conduction only occurs in myelinated neurons, in neurons that have myelin. If you are an unmyelinated neuron, if you're a neuron that does not have myelin, you will do continuous conduction. And then we talk about things that could affect the speed of the conduction. How fast can it go? Well, there are a couple things that could affect the speed. One, we kind of mentioned, has to deal with whether or not you have myelin, because we already know if you have myelin, you're going to do saltatory conduction. So you're going to go faster. Okay. But also, the size of the axon matters. Yeah. Turns out, if you have a large diameter to your axon, you're able to conduct it faster. But if you have a smaller diameter, will go slower. Again, think about being in your car. Imagine, where do you think you'll be able to go faster? Can you go faster on a 10-lane highway, which Houston, some reason, has 10-lane highways? Or do you think you'll go faster on a 2-lane road? No, you're going to go faster on the highway. Same thing. If you give things more room, they could go faster. The bigger diameter axon will have faster conduction than the smaller diameter axon. And the axon with myelin will go faster because it will do saltatory conduction than the neuron without myelin. That's doing continuous. So those are two major things that affect the speed. How big is the diameter of the axon and whether or not you have myelin. And then we talk about the end result. You know, the action potential is going to eventually arrive at the axon terminal. And we saw in a muscle contraction that will lead to the axon terminal releasing acetylcholine to get your muscle cell to contract. And it happens at a neuromuscular junction. Yeah. Turns out a neuromuscular junction is a type of what we call a synapse. A synapse is just an area, a place where a neuron will talk to another cell, where it will meet, innervate, excite, talk to another cell. Okay. So we got to know the parts to a synapse. You know already because you know the parts to a neuromuscular junction. And a neuromuscular junction is an example of a synapse. So you know one part of a synapse will involve the axon terminal of the neuron sending the signal. And remember, the neuron does not need to touch the cell it talks to. There's a synaptic cleft. That's the second part. And then the third part is the plasma membrane of the cell receiving the signal, which could be a muscle cell, but it could be another cell. Neurons could talk to other neurons. So it could also be the plasma membrane of a neuron receiving a signal. Those are the basic parts to all your synapses. The axon terminal of the neuron sending the signal, the synaptic cleft in the middle, and the plasma membrane of the cell receiving the signal. And I told you neurons could talk to other neurons. So we even classify synapses based off where those neurons could talk to each other. For example, the axon of one neuron could talk to the dendrite of another neuron, which is what you see here. So we call this an axal dendritic synapse. Just break down the names. Axal for axon, dendrite for dendrite. It's telling you what's talking. Or I could have the axon of one neuron talk to the cell body or the soma of another neuron. That's an axosomatic synapse. Or I could have the axon of one neuron literally talk to the axon of another neuron. That's an axoaxonic synapse. Remember, don't be intimidated by names in this class. They tell you what's going on. Okay. And we could classify synapses based off how they're sending the information. You could send the information with electricity. If you use electricity at your synapse, then that's an electrical synapse. We saw that in things like gap junctions, okay, where cardiac cells send electricity via those gaps to another cell. That's technically electrical synapse. Or you could use chemicals to send a signal, like what we saw in the neuromuscular junction, where we released our chemical neurotransmitter, acetylcholine. Well, in that case, you have a chemical synapse. Turns out chemical synapses make up the majority of synapses in the nervous system. So when we saw the neuromuscular junction with the neuron releasing acetylcholine, we were looking at a type of chemical synapse, to be specific. All right. So since this is majority of the synapses in our body, we got to look at how does it work. Okay, back to your neuromuscular junction. We saw acetylcholine was trapped in a little bubble we called the synaptic vesicle. Here it is. Here's our axon terminal. We see these little circles inside. That's a vesicle. Remember, those are fluid-filled sacs that contain chemicals inside. Well, the chemical in this case are our neurotransmitters like acetylcholine. We got to see, how do you release it? Just think back to the steps of our action potential. Think back to the steps of our muscle contraction. Remember, it began with an action potential arriving at the axon terminal, and I told you that triggered the opening of voltage-gated calcium channels, because that's what happens in a chemical synapse. And once you open these voltage-gated calcium channels, remember what happened next? Calcium rushed into the cell. And when it rushed into the cell, that's what triggers the release of your neurotransmitter, which will then diffuse across the synaptic cleft and bind receptors on the muscle cell's plasma membrane. You are seeing just the events of you release a chemical in a chemical synapse. Look at your exam review. I was just giving you that in chapter 10. Action potential arrives at the axon terminal, triggering the opening of voltage-gated calcium channels. Calcium rushes into the cell, triggering the release of acetylcholine. I was explaining a chemical synapse. So look at that. Okay. And once that neurotransmitter binds to receptors on the cell receiving the signal, which can be another neuron, in that case we call it a postsynaptic neuron, that cell will then possibly generate its own action potential, like we saw with the muscle cell generating its own action potential. Look at that. We know all this already. All right. What else? Let's go back to chapter 11. So we know what happens at a chemical synapse already. And then once that cell receives a signal, I told you it could potentially generate an action potential. So we got to look at the results of a cell receiving a signal. Yeah. Turns out the cell could generate either an excitatory postsynaptic potential or an inhibitory postsynaptic potential. These postsynaptic potentials are just slight changes in the plasma membrane's charge difference. They're what we call local potentials. Okay. The difference is, are you getting pulled towards or away from threshold? That's the major difference between excitatory postsynaptic potentials and inhibitory ones. So let's think. If I get a signal to do something, like my muscle cells get a signal to contract when they get acetylcholine binding to the receptors, you're going to get an excitatory postsynaptic potential. Kind of think you've been excited to do something. And in order for you to do that, you need to generate an action potential. Remember, the first step is hitting threshold. So whenever you generate a excuse me, an excitatory postsynaptic potential, you're getting pulled towards threshold so that you could do depolarization and make your potential. So you tend to see this when you get an excitatory signal. But sometimes some neurotransmitters tell you to stop doing something. Sometimes you get an inhibitory neurotransmitter. And in that case, you're going to generate an inhibitory postsynaptic potential. How? How do you get a cell to stop doing something? Well, you cause it not to generate an action potential. How? You keep it from threshold. How? You pull it away from threshold. How? You make it more negative. Okay, you force it into a small hyperpolarization. That is your inhibitory postsynaptic potential. You hyperpolarizing, so I pull you away from threshold so that you cannot do something. So those are differences in the results you're going to see once a neurotransmitter binds to a receptor on a cell receiving the signal, like a postsynaptic neuron. What else? Now you know the results you can see. So now we could complicate it. Oh boy. We know some neurons can get excitatory signals, generate an excitatory postsynaptic potential, bringing them closer to threshold. And we know some neurons can receive an inhibitory signal, pulling them away from threshold by hyperpolarizing them, and what we call an inhibitory postsynaptic potential. But I mentioned we're going to complicate it. Turns out one neuron can actually receive multiple mixed signals. A neuron could receive some inhibitory signals and excitatory signals at the same time. So what does the neuron do? This is like me telling you to go study right now, but also me telling you to not study at all. What do? Do you study or do you not study? I don't know. Turns out if you're a neuron, you're going to do both. And neurons do what's called summation. Kind of thing. Summation, you're summing things up, you're adding things together. This is your neuron listening to all the signals. It's going to add them up. Okay. And it has different ways to do it. There are different types of summation. There's temporal summation and there's spatial summation. They are both summation, so they are both your neuron adding all the signals it's getting. The difference is in the name. Temporal for tempo, so think time. Okay. So this is when a neuron listens to and adds up all the signals it's getting within a certain period of time. All right. But on the other hand, a neuron could listen to and add up all the signals it's getting within a certain area, certain space. So we call that spatial summation. That's what you see in these two pictures here. Over here on this side and under the A section, that's temporal summation. Over here in the B section, this is spatial. In temporal summation, you can listen to all the signals you get, no matter where you get them, within a certain period of time. So if it's an excitatory postsynaptic potential, you'll listen to it, move closer to the threshold. If it's an inhibitory, you'll move further away. It's just over time, and by the end, you see where you are. In spatial summation, you're only listening to all the signals you get in one area. Like in this picture, they're listening to all the signals they're getting in this one area of the soma. That is spatial summation. Doing the same thing. Excitatory, bringing you closer to threshold. Inhibitory, pulling you away. And once you're done listening to everyone in that area, then you see where you are by the end. Whole point is, did I hit threshold or not? If I hit it, I will make my action potential. If I do not, I won't. So yeah, if I tell you not to study and to study, well, you can listen to how many times I say both. Wherever you are by the end, well, you'll do it. Or you could listen to me telling you all the things to do in one specific area. Okay? And that's the difference between temporal and spatial, time or space, literally. Mhm. But then let's say you want to stop sending a signal. Okay, remember, for you to move, you need to release acetylcholine. But what if I don't want to move? What if I want to stop sending a signal? I need to remove my neurotransmitter. Turns out there are 3 ways to get rid of a neurotransmitter. You know one of them already. Think about how you got rid of acetylcholine. But turns out there are 3 ways in which you could get rid of a neurotransmitter, and you got to know all 3 ways. There's something called enzymatic breakdown. We saw that. It's literally when an enzyme breaks down the neurotransmitter. Think acetylcholine esterase breaking down acetylcholine. That's one way. Another way is called diffusion, where your neurotransmitter literally diffuses away from the synapse. It literally walks away from the synapse. Or you could do something called reuptake. This is when the neuron that released the neurotransmitter literally sucks it back up. It reabsorbs, takes it back. That is reuptake. We see all 3 here. You can have your neurotransmitter literally leave the synapse. It just happens to diffuse away, going from an area of high concentration to low. That's diffusion. It left the synapse. You could break it down with an enzyme. That's degradation. Or you could have the neuron that released it take it back up, suck it back up, reabsorb it. That is reuptake. No matter what, they all help you to get rid of the neurotransmitter in the synapse so you stop sending your signal. And just know that acetylcholine is a major excitatory neurotransmitter, but it's not the only one. There are lots of neurotransmitters. It's in a little chart in your textbook. Let's go to that chart. Or in PowerPoint, sorry, which is also in your textbook. Oh, where's that chart? Oh, here we go. So you can imagine your neurons do a lot of work. And to do every specific thing, you'll need different neurotransmitters. So this table gives you a little list of just a few neurotransmitters. Some of them are excitatory, so they'll tell cells to do something, while some are inhibitory. Turns out acetylcholine, it excites muscle to do a contraction. Why? Because it's an excitatory neurotransmitter. But there are some inhibitory ones. There are some neurotransmitters that will get cells to stop doing something, like GABA. GABA is GABA. That's an inhibitory neurotransmitter. Or there's some neurotransmitters that send specific information, not just excitatory and inhibitory. It literally carries specific information, like substance P helps to send pain signals. Well, yeah, when you stub your little pinky baby toe on the side table and life feels like it's over because you're in so much pain, you're feeling that pain thanks to substance P. So some neurotransmitters are excitatory, acetylcholine, some inhibitory, GABA, and some send specific information like pain. Think substance P. Kind of think the P in substance P is for pain. And then to finish off, we talk about healing a neuron. Oh, Dr. Higgs, I thought you said nervous tissue fibrosis. It does. Yeah. But there is a small area where you can regenerate. Yeah. You got to know this one line. Your neuron can regenerate, but only in one instance. You can only regenerate a neuron if the cell body remains intact. So that means you could really only damage the axon or the dendrite. If you damage the cell body, it will not regenerate. So let's look at it. How do we heal and regenerate a neuron? Really, how do we heal and regenerate anything but the cell body? Because if you damage the cell body, it's not going to regenerate. Let's go through that. So let's say you're Homer Simpson and you're in the kitchen with Marge. She's cutting up a pie or some brownies and you reach for it and she cuts off your thumb. Now you got to rush to the hospital for them to sew it back on. That was an episode once. Turns out if you get to the hospital in time and they sew it back on, you might potentially be able to resume function in that finger because you'll be able to move it again because you regenerated the nerves that were cut through. How? Well, turns out your body goes through some steps in the regeneration of a neuron. Sorry, I was looking for the picture. And it's a little counterintuitive, the order of neuron regeneration. The steps are what we call Wallerian degeneration. Then you're going to have growth of the axon. Then you're going to have a regeneration tube formation. And then you'll finish growing the axon until it attaches to the part that it was severed from. It's just 4 basic steps. Okay? Wallerian degeneration, chromatolysis, that's step 1. Growth of the axon, that's step 2. Formation of your regeneration tube, that's step 3. And then you'll finish growing the axon and connect it to the target cell that it was separated from. That's step 4. Sorry, I was looking for the picture. But that's it. First step is Wallerian degeneration and chromatolysis. When you cut that axon, there's the part that's still attached to the cell body, and there's the part that's reaching towards your target cell, the cell the neuron was originally talking to. You would think, well, your body will just simply reattach those 2 areas in the middle. It doesn't. When you damage that axon, it breaks down the distal part of the axon. It breaks down the part of the axon that was closer to the target cell. And it will simply just grow the axon from the part that's still attached to the cell body. When you break down that distal part of the axon, that is Wallerian degeneration and chromatolysis. It's just you breaking down the part of the axon that was cut away from the cell body. Why? You don't need it. Step 2, you're going to take the axon that's still attached to the cell body, and it's going to sprout and begin to grow, kind of like a plant. But you could imagine it was cut off from its target cell, so it doesn't know where to grow towards. It needs help. It needs guidance to know where to grow towards. That's when Schwann cells are going to come and form a tube made out of myelin. We call it the regeneration tube. That's step 3. Once the Schwann cells form the regeneration tube, now the axon will go into step 4, where it will continue to grow, and it will grow through the regeneration tube until it reconnects to the target cell. And that's how you regenerate a neuron. So Wallerian degeneration, chromatolysis, break down that distal part of the axon, step 1. Step 2, begin to grow the axon. Step 3, form your regeneration tube with your Schwann cells. And step 4, finish growing the axon through the regeneration tube until you hit your target cell. And those are the steps in the repair or regeneration of a neuron, as long as the cell body is still intact. You have not damaged the cell body. And that's nervous system. That's chapter 11. And that's your exam review. Take your time. Like always, if you have questions, just let me know.